In this video, we will go over the Physio Ex8 assignments that you had last week. And in order to understand what was going on with the assignment, we will establish a few facts. The first one is the reaction itself that we had in the assignments. The basic reaction was that the starch in the presence of amylase will give us glucose. As you know, this is polysaccharides, and the glucose is a monosaccharide. Amylase is an enzyme that breaks carbohydrates. And in our example, it's going to break starch to monomers. This is the basic reaction we had in the assignments. And you already know this, or you should have remembered it, that each enzyme has ideal conditions to work with. Basically, what we tested in the assignment, we tested the pH and we tested the temperature. For the pH, there is an ideal range of pH that should be around 7 for this enzyme to be working, and for the temperature, it should be around 37 degrees Celsius. So any changes in these values, either up or down, it's going to affect the enzyme activity. So basically, that's what you tested. You tested how the changes in the pH and the temperature is going to affect the amylase activity. And how do we measure the amylase activity? By looking at the presence of glucose in the tubes. Now, how did we understand, or how did we figure out that there is glucose at the end of the reaction or not? If you remember, in AB1, we talked about something called Benedict's solution. Benedict's solution is going to change its color from blue to red in case there is sugar presented. So Benedict's solution acts as something we called indicator. So if there is a change of color, I will know that there is a monosaccharides in the tube. Now the other indicator was used in the experiment was something called the IKI, and this is basically iodine ions. The IDI, usually it's red, but in the presence of starch it will turn into blue. Technically it will turn the color of the starch into blue. So that's the second indicator. So we have the Benedict solution for the glucose, and we have the IKI for the starch. So let's go through the experiment. You had several tubes. So let's see what was going on with these different tubes. We had 8 tubes. 1, 2. In each tube, we had different mix of substances. We had amylase, we had starch, and we used water, the ionized water. Also, we used maltose. Also, we looked at the temperature, and we looked at the pH. We changed the temperature sometimes, and we sometimes changed the pH. For the first tube, it has amylase and it has a starch. It did not get any water. It did not get any maltose. Temperature was boiled and pH was 7. So this was the first tube. Second tube had also amylase, and also starch, no water, no maltose. It was frozen and pH was 7. Third tube, also amylase, starch, no water, no maltose, no temperature treatment, and pH was 7. 4. It has amylase, but it did not have starch, and instead it got water, no maltose, no treatment in the temperature, and the pH was 7. Tube number 5. It did not get amylase. It has a starch. It did get water instead of the amylase, no maltose, and no treatment, and pH is 7. 6. Also no amylase, no starch. It got water. It got maltose, no treatment in temperature, and pH of 7. Tube number 7. It has amylase, starch, no water, no maltose, no treatment of temperature, but the pH was 2. And 8 as 7, amylase, starch, no water, no maltose, no temperature treatment, and the pH was a little bit alkaline. So that was the experiment. Now let's go through what expectations you would have. So we will have the Benedict solution. Origin color is blue. And we are going to check with the IKI. Origin color is red. Remember, Benedict solution is going to tell us about the presence of glucose or a simple sugar, whereas the IKI will tell us about the presence of starch. So let's see how Benedict solution is going to act in these reactions. And remember also, before that, Remember that we need ideal temperature, we need pH, and the reaction is starch in the presence of amylase will give us glucose. So we need to keep in mind all of these when we are checking the results of the tubes. So for the first tube, we have amylase, we have starch, but the temperature is boiled. So the enzyme in high temperature, it will get broken. You know, enzymes are proteins. In high temperatures, will get degraded. So they're ineffective or not active anymore. So in this case, do you think that we will have glucose in the tube? We will not. So the Benedict solution would remain blue. It will remain blue because the amylase is inactive or was destroyed. So therefore the starch remained as starch and wasn't converted into glucose. So in this case, AKI, in the presence of starch, it will turn to blue as well. So in this tube, in the first tube, we would know that the reaction did not work because there was damage to the enzyme because of the temperature. On the second tube, the same components, however, there was a freezing temperature. The enzyme was frozen before carrying on with the reaction. And technically, freezing the enzyme is not going to affect its nature. So the enzyme is still effective. So in this case, in this tube, we will see some conversion of starch to glucose. Okay, so the Benedict solution will turn from blue to red, whereas there should not be any starch left in this tube, since the enzyme is going to convert all the starch to glucose. So the AKI will not find any starch to change its color. So the AKI would remain red. For the third tube, amylase there, starch there, no water, no maltose, no temperature in the treatment, the pH is ideal. So this is like a control tube. We know here that we will get red in the Benedict solution because all of the conversion happened to glucose and there should not be any starch left, so it will be red. Okay, so this is our control. In tube number 4, we had amylase, but there is no starch. And instead of starch, we put water, and we did not change anything else in the temperature and the pH. So in this case, amylase, there is nothing for amylase to work on. There is no starch, there is nothing. So in this case, for the Benedict solution, it will remain blue because there is no glucose, there is no end product. And the AKI would remain red. Why? Because there is no starch to start with. Tube number 5 is the opposite. We did not put amylase, we put the starch and we put water. And we did not change the temperature or the pH. So here, for the Benedict solution, it will remain blue because there is no glucose. But the AKI, it will turn into blue. Why? Because there is a starch, and that starch remained in the tube since there is no enzyme to convert. Tube number 6, there is no enzyme, there is no amylase, there is no starch. It's only water and maltose. And maltose is a form of simple sugar. So with this tube, Benedict solution is going to turn red because of the presence of the maltose. AKI would remain red. Why? Because there is no starch to start with. Now, 7 and 8, we change the pH. There is amylase in both, there is a starch in both, no treatment in temperature, it's only changes in the pH. And for the changes of the pH, it will affect the effectiveness of the enzyme. So you would not expect a full conversion of the starch into glucose in both tubes, because this is extreme acidic and this is a little bit alkaline. And technically, with pH as low as 2, we should not expect too much of conversions to the glucose. So I will say, instead of red, it will be like dark red or purple. Like there is some conversion, it indicates some conversion, but not as effective as the control. Similarly, for the AKI, there will be some blue color. Like we can think of light blue maybe. There is some presence of starch remained in the tube. And this is also the same situation here. There is some dark red and there is light blue. So basically this is the experiment and this is the interpretation of this experiment. That you need ideal conditions in temperature and pH. You need the presence of the enzyme and its substrate. And what you are going to see in the exam, or what you should be able to do in the exam, that you will be given the results and you need to explain these results based on the information provided for each tube. So if it turns blue, that you would know that Benedict solution detected some monosaccharides. If the IKI remained turned blue, you would know that the IKI detected some starch, and so on. Now we said this is the control. Now we have also a positive control. So we can think of this as a positive control and these two as a negative control. Positive control, it means that you are going to expect to see the change as they should happen. Whereas the negative control, you know that it's not going to work. That's why it's negative. It's not going to work. In this case, in tube number 4, you put the amylase, but you did not put the starch. So you want to see here if the amylase by itself is going to change the colors of the indicators. Okay? And the same thing for the question 5. The amylase is not there, but the starch is there. And you want to see if the starch is going to affect the color of the Benedict solution. So if you figure this is correct, or the controls are working as they should be working, you would make sure, or you will be sure that the results of your experiments are okay. You will not have any doubts in the experiment. I hope it's clear. Let me know if you have any question.